Hello everyone, in this video I'll be explaining on the topic of the applications, advantages and limitations of phase contrast microscope. This phase contrast microscopy is an optical microscopy technique that converts phase shifts in light passing through a transparent specimen to brightness changes in the image. These phase shifts are invisible but become visible when shown as brightness variation. It was described in 1934 by Dutch physicist called Fritz Zernike. Phase contrast microscopy improves the contrast and makes evident the internal structures of cells which differ in thickness of refractive index. The different part of a cell and its surrounding medium have got different refractive indices. These unstained living cells absorb practically no light. When poor light absorption results in extremely small differences in the intensity distribution in the image. This makes the cells barely or not at all visible in a bright field microscope. A light microscope can be converted into phase contrast microscope by using a special condenser and objective lens. Coming to the applications of phase contrast microscope, this is used in microscopy observation of unstained biological materials such as the living cells, which are usually done in case of culture, and then it is used for viewing microorganisms, thin tissue slices in case of histopathology or tissue examination, and subcellular particles including the nuclei and organelles of a cell. So these are the various applications of the phase contrast microscope. Coming next to the advantages or the positive points of phase contrast microscope. Living cells can be observed in their natural state without previous fixation and labeling, where because in some of the cases where you need to observe various microorganisms or cells where it needs to be prefixed or pre-stained. But here in case of phase contrast microscope, living cells can be seen where with their natural in their natural state where no fixation or labeling is required. Next is it makes a highly transparent object more visible. That means there are some structures which are very thin which can be seen by under normal light microscope, or they become too transparent, right? But here in phase contrast microscope it can also see such kind of structures which are highly transparent under normal ordinary light microscope. Next is no special preparations or fixation of stain is needed to study an object, which saves a lot of time. That means in most of the cases where observation of a particular microorganisms or cells need a prior fixation or a prior staining so that the particular organisms or cells can be viewed under the microscope. But here in case of phase contrast microscope, this process need not required for observing under phase contrast microscope, which saves a lot of time because in those methods where staining and fixation is required, it undergoes various steps and process. But here in phase contrast microscope, the observation need not be fixed or preparations or staining need not be done, but It can be observed without them, which saves a lot of time. Next is examining intracellular components of living cell at relatively high resolution. So all those extracellular components of a cell can also be viewed or observed under this phase contrast microscope with high resolution. So this is the advantages of phase contrast microscope. Next, coming to the limitations of phase contrast microscope. Here the phase contrast condenser and objective lenses add considerable cost to a microscope, and so phase contrast is often not used in teaching labs except phases perhaps in classes in the health professions. To use phase contrast, the light path must be aligned. Generally, more light is needed for phase contrast than for corresponding bright field viewing, since the technique is based on the diminishment of the brightness of most objects.